Thank you very much for this kind introduction and welcome everyone that has joined us today. What I would like to start with are the challenges in molecule discovery. And one of the most significant issues that we have as chemists is the complexity of all of the chemical transformations that are there. So there's actually no single individual that can remember all of these reactions that have been performed before. And this number is actually constantly growing, right? So there might be the case that for your target molecule, there might be a transformation that would allow for its synthesis in just like few steps, but sometimes identifying this simplifying step is very challenging, especially when it involves less common chemistry type. On the top of that, chemists are under pressure, right? We want to synthesize new molecules, bring the new drugs to the market as fast as possible. So this urgency adds another layer to the complexity of this process. When working with a published reactions or published targets, we often find that the literature route is not reproducible. So it might be the case that you try to perform a particular literature pathway in your lab and it actually doesn't work. While on the other hand, you're working on a target that is completely novel molecule, never synthesized before, like for example, new drug candidate, there might not be sometimes even a similar synthesis in the literature. And furthermore, a failure of the synthesis can happen actually at any step. So it would be great to have something that really provides multiple options of how you can make your molecule. And finally, it's really impossible to remember the entire catalogs of molecules that you can buy. So it would be great to have something that really unites all of this together. And this is actually why SYNTHIA was designed. These are the challenges that SYNTHIA can help you to overcome. So we created this solution to not only predict synthetic pathways for individual molecules, but also to optimize the synthesis, to help creating new libraries of molecules, and many, many more. So basically, what SYNTHIA does, it supports chemists in their daily work. So you can, for example, use our similarity searches to check the commercial availability of your molecule, or you can generate really big libraries of new compounds, analogs of your targets, using our forward synthesis algorithms. And I would like to show you today a couple of those advancements. But first, to start with, it's very important to just at least even quickly go through the key pillars of SYNTHIA, what the software actually does and how it does it, so that basically all of these results that we will be showing today will be understandable for you. So what you can see right now on my screen is an example of a reaction rule. Reaction rules database is actually at the heart of our SYNTHIA software. And what it does, it helps to create synthetic pathways, right? SYNTHIA uses these rules to create the synthetic pathways. And these rules are created by our expert PhD level organic synthetic chemistry team. And how also they are written is that they need to consider all of the things that actually human chemists would consider when looking at the reaction. Like, for example, what are the steric effects that influence the creation of a particular target? Or, for example, what are the electronic effects that you need to take into account in a particular rule? Or, for example, what are the incompatible groups, so functional groups that would not survive these reaction conditions, and many, many more. But also, these rules are written in a general way, so that allows them to be applied to really known molecules published before, but also on the completely novel targets that have never been synthesized before. And this is an example of just one of such a rule. This code here describes all of these crucial aspects that needs to be there in the structures of the substrates for this reaction to proceed like that, but also all of this context informations like, incompatible groups or literature references or reaction conditions. And the second part, pretty important as well, are the algorithms that are responsible for chemical logic, right? So this helps SYNTHIA to design the pathways that are selective, not having any strain-related problems, or, for example, are not having any structural motifs that would be unstable, right? Or that would, for example, undergo rearrangements under certain reaction conditions. So it assures the chemical correctness of the predicted molecules and predicted reactions. And this algorithmic part has another side as well, which are the algorithms that are responsible for strategizing over multiple steps during this pathway creation. So, for example, SYNTHIA can combine the reactions that can be performed under the same reaction conditions, or those that once applied can really provide a significant structural simplification, or are stabilizing, highly reactive groups along the synthetic pathways. But what is very, very important here to say is that all of these algorithms in SYNTHIA are working together, analyzing these pathways globally, analyzing all of these millions of possibilities of combinations of reactions to come up with a solution that is the most optimal. And also, let's now move to the third SYNTHIA pillar. And this pillar actually is the advanced path search algorithm itself. And what this algorithm does, it's scoring and exploring all of these possible connections, combinations of reactions, and putting them into full synthetic pathways that can start from something that is commercially available, and also taking into account all of the requirements that are coming from the users, right? So we discussed chemical knowledge, so this expert coded rules that are super important for the prediction quality and also diversity of the results. We talked about chemical logic that is responsible for providing and creating chemically correct molecules and chemically correct pathways. We talked about search algorithm that is combining all this together, but the last very, very important thing, which is a pillar 4, is the customizability of SYNTHIA, because every synthetic chemist and every synthesis can really present different demands, right? So, for example, some customers can use metal catalysis while others cannot, right? Some chemists are, for example, interested in carbon footprint information for their pathways, while the others would like to, for example, scale up the synthesis. So they would like to start with starting materials that are available in like bigger quantity. Some chemists would like to start with chiral pool molecules and many, many more. So we designed SYNTHIA to allow users to easily specify additional constraints for their synthesis, which also makes the results adaptable to really diverse needs of the users. Let's now move quickly to how actually SYNTHIA presents the results. And here you can see my homepage of SYNTHIA. Here I have my analysis I performed before. And if you click on this new analysis button, what you will see is actually, the drawer window in which you can put your molecule, you can draw it, you can paste it as well. And the last thing actually that remains is to just click start button. What SYNTHIA does then, it's starting looking for the pathways and show the users the results. So let's take a look quickly on how this looks on a short video. I clicked on the new analysis button, and right now I will type a part of the name because I want to synthesize a drug that has already been published. SYNTHIA is right now searching for our published molecules database, and then you can select one of any variations of these molecules that are there available in our database. By selecting one of them, it just appears in our drawer. You can change the predefined search conditions, but for this search, we are going to go with general and just click start button. So what SYNTHIA currently is doing is searching for millions of potential ways of how you can actually make this molecule, right? So it's really traversing a very big graph of possibilities. So also maybe to answer the question of how long such a search can take, it depends, of course, on the complexity of the molecule. For smaller molecules, it can take a couple of seconds. For less complex one, if this is something very complex, like a very complex natural product, it can take up to a couple of hours. And as you can see, since you already found the results, and it will keep searching, right? Keep searching for the results. as you see, computation is in progress, and whenever Cynthia finds anything, there's this bell icon you can click on to refresh and download new results. And this is basically how Cynthia presents the results, just like, as a default view. This is a building blocks view, but there is, of course, a pathway view, and I would like to show you how you can review the results in Cynthia in a couple of next slides. What you can see here are a couple of ways of how you can review the results provided by Cynthia. We have bonds disconnection view, the building blocks view, the pathway view, and as well the graph view. So I would like to show you this as well on a second video that I have. And what you can see as well here, this is my dashboard of analysis I performed in Cynthia, right? So there are many of those. There's always something at the front of the analysis. And we will just take a look at one of these analyses I ran before for sitagliptin. And this is a default view of how you can see the result. This is called building blocks view. And as you can see, there are these colorful parts of the building blocks that corresponds to these colorful parts on the target on the left, right? So you see basically which one of these building blocks parts constructed which part of the target. And as well, it's very interactive. You can click on the molecules. You can take a look some more details. And we go to the second view by clicking on the target. And here you can hover over the bonds that Cynthia proposed to create. And you can see the names of the reactions that Cynthia proposed in this particular pathway. And now we'll move to the third view by switching of this toggle to the pathway view, so something which chemists are most used to. Let's just zoom into it a little more. And what you can notice, there are, of course, names of the reactions, conditions, and molecules. And this is very interactive, so you can as well click on everything, on the name to see the typical conditions, to see the illustrative references, to see as well the similar reactions that were published. They're similar to the one that Cynthia predicted, right? We have a couple of published reactions databases, and everything is clickable as well here. So you can click on the book icon, for example, to just directly take a look at the literature, similar reactions to this one that Cynthia predicted, and just you can navigate like that. And also the molecules here that are on this pathway are also clickable if you would like to get some details about the prices, about their references, or any links to PubChem, for example. And Cynthia by default gives 50 pathways that you can filter down, narrow down. You can download them in PDF, as an image, and many, many more things you can do with this view. So Cynthia provides you reaction conditions and names. It provides you similar reactions from the literature, and as well protecting group informations, PubChem links, and also safety and sustainability information, which is very, very important if you are trying to optimize your synthesis in the context of green chemistry, right? So all this information can really support you in selecting your final synthetic pathway. And also there's one last view, which is called graph view, which is an overview, global view of all the pathways that Cynthia proposed, that you can also narrow down and filter from this view directly. What is also a pretty recent thing that we've added is the database of enzymatic reactions that can also help you in creating more sustainable pathways for your targets, using the reactions, the rules that are catalyzed by the enzymes. And one other algorithm that Cynthia has, and this was recently added, is the algorithm that predicts chiral separation steps in the pathways. And Cynthia identifies here the structural motifs that can be separated and includes such motifs and such reactions as steps in the predicted pathways. And now I would like to talk about something that is really brand new. This is a completely new Cynthia module, which supports chemists in multi-target oriented analysis. And I will be talking first about diversity library, and then my colleague Emma will talk about another multi-target oriented analysis feature, which is called shared path library. But let's start with this diversity library one. So what it does, it enables to automatically design target libraries based on the synthetic pathway. So what the user does here, he selects starting materials, which he or she would like to diversify. And with this selection, Cynthia generates a library of targets that are similar to the original one. So it might be the case that these molecules have never been published before, that they are completely novel, never synthesized. But what is extremely important to say here is that Cynthia generates this library considering its entire power of the reaction rules, with all this context information, with all the incompatible functional groups in every reaction, and this entire power of algorithms as well, right? So the algorithms that we have talked before about, removing non-selective reactions, non-existent motifs, and also other algorithms that we have built inside our Cynthia tool. So all of this together means actually that the library of these targets that the software generates is really chemically correct. So the structures are chemically correct and are synthesizable. And I would like to show you that as well, how it works on a short video. And here's once again my Cynthia dashboard. I will click on one of this analysis, and this is on a Capavir, very recently published drug treating AIDS. And this is this building block view, but there's this icon, Create Diversity Library, right? So let's click on that together. And what you can also see here, there are these boxes for selection of the building blocks that you would like to diversify. Right? So you are right now selecting the points of diversification that will be different in this generated target comparing to this original one, right? So you are as well, you are guiding Cynthia to what diversity you are looking for, and then you select the number of molecules that should be generated, right? And with that selection, what Cynthia does, it creates you a library of completely novel molecules, right? Completely novel targets. And what you can also notice here, there are these yellow parts, right? And what this means are actually the points of diversification, right? The differences between this original target and this targets that are generated here, right? And you can review this results in many ways, but what is very, very important to say is that every of this molecule comes as well not only as a structure, but with a full synthetic pathway that was used to generate this molecule. So you are getting as well the process of how to make this molecule in the lab. What you can also do here, you can sort these results differently by, for example, similarity to this original target, which is in the frame, blue frame, or by the molecular weight. And you can also color them differently, like, for example, based on building blocks. And with that selection, what you will notice, there are these different parts of your molecule, which really enable easy spot of the differences between the generated targets. Let's take a look at, for example, 12 and 13. There are defluoromethyl group, here's trifluoromethyl group, and many different diversities you can notice here in the generated molecules. And let's go right now to how you can download these results and what you can do with these results. So once again, we are in the results for diversity library. What I'm doing right now, I'm selecting the molecules that I would like to download, and you can select every molecule, or you can select like only a couple of those. I selected 8 molecules, and you can download them on many different ways. You can download them, for example, as images, but also as CSV file with SMILE structure. So you can directly then take these molecules and load it, for example, into your drug discovery tool for further processing or further filtering if you would like to do anything else with this molecule afterwards. And now I would like to pass over to my colleague Emma, which will lead the second part of this webinar.
Great, thanks, Ava, and thanks, everyone. I'm going to go ahead and share my screen here. All right, so for the second half of this talk, I'm going to go through some workflows and applications using SYNTHIA. So to get started, if we look kind of at the big picture of the drug discovery and development workflow, we start at the beginning with biology research, identifying a target that we want to modify, and then we move into the what to make phase of determining what molecules are going to have the desired effect on our biological target. Once those are identified, then we have to figure out how to make them, and this is that chemistry route planning. And then finally, in the last stage of actually doing the synthesis, and then once we've identified our lead target, optimizing that and doing the scale up. And so SYNTHIA really kind of sits in the middle here, supporting that how to make route planning phase. But what I want to show is we actually have features that can really be applied to across this spectrum, both to that early discovery research of looking how to make that, or I'm sorry, what to make, as well as the later stage, more process development research. So starting off, if we kind of zoom in on that discovery phase, typically here we have the design, make, test, analyze cycle, where we identify some compounds that should have desired properties. Then we've got to figure out how to make them so that we can test them in assays to assess those properties, and then analyze the results, which then informs a new round. And how quickly we can iterate through that really drives our discovery process and making that more efficient. So SYNTHIA can really accelerate the make portion of this by helping you to decide how can I make the most compounds quickly, maybe what compounds to prioritize making, and identifying what starting materials you can buy. And so we actually have a feature specifically designed for this that Ava mentioned called the shared path library. So now instead of looking at optimizing the route for a single target, we have a library of target compounds that we want to make, and SYNTHIA looks at how we can optimize the synthesis for the entire library. So it's going to prioritize convergent routes that have shared synthetic steps and common intermediates. So if you look at what I have on the screen here, I've given it this sort of simple library of these 4 compounds. SYNTHIA's found a route that's 3 steps. The first 2 are shared for all 4 targets. So you can see we've got this shared penultimate intermediate here, and we've got the little 4 indicating that we can go through that to make all 4 of our targets. And then in the last step, we're doing the Suzuki coupling with 4 different boronic acids to access the 4 targets. So this is really useful to be able to find convergent routes to synthesize your library, identify commercially available building blocks, and maybe prioritize what compounds are going to be easier to make. And so to look at that in a little more detail, I have a video here. So we start the same way as we set up a normal search, but now we may want to upload a file of structures. So it takes a mol file or an SD file. Once you load it in, it's going to pull in all your structures and automatically recognize that we are doing a multi-target analysis and switch over to the shared path library. We can still customize the search just like you normally would, and then hit start there. And so then once the search finishes, instead of a bunch of pathways for one target, I've got a set of pathways, 1 for each target, and it gives you the summary. In this case, I've got 26 unique reactions to make all 12 of those compounds. And then you can see the pathway for each target listed here. We can go ahead and kind of take a look at the details here. And again, we've got a late stage diversification. So the first 3 steps are shared for all 12 targets. And then in the last step, we're doing Buchwald-Hartwig amination. And for this target with a commercially available amine here to get to the final product. We can take a look at the other targets, here's another commercially available amine, but here's one that has to be synthesized in one step. And another one that requires a one-step synthesis to get to that amine. In this case, you can see that the final amine actually is commercially available, but quite expensive, so it shows you how to make it from something cheaper. So in this way, we can kind of see which of those targets are going to be the quickest to make because they're the amines commercially available versus some that, here's one that requires 2 steps. So this can help you to prioritize maybe within your library, which ones are going to be the easiest to make. And then we can actually take this one step further and use that diversity library that Ava showed right from the shared path library and see, maybe there are some other targets that would be easy to make that are similar from other commercially available amines here. So I'm just going to go ahead and diversify right at that position and create a new library. And now you can see Cynthia's found 48 targets, all with reasonably high similarity to our original target coming from commercially available starting materials there that might be easy to access. So if we click on one of these for example, I can see here's an amine that's relatively inexpensive as well. So this is sort of one example of how you can use these multi-target analyses to help you iterate through that DMTA cycle. And so now if we kind of move through the process and jump ahead now to more of that production development stage of now we want to optimize a single synthesis. We have some other priorities there, wanting to have the most efficient route for our lead target. And one of the things that we're often looking at is how we can make sure that our process is as green as possible. And this is something that Cynthia can really support, those sustainability goals. One of the things kind of built into the algorithms is looking at atom efficiency. So it's going to look for routes that have high atom efficiency over less so. One of the ways it does this is by avoiding protecting groups, which can also, save you steps and make that route more efficient as well. And then looking at regulatory considerations and really giving you information that you need to inform decisions about whether you want to use something or chemicals that may be greener. And so we do have some sustainability information kind of right there, make it easy for you to find. So you'll notice right on the building blocks themselves, in this building blocks view, you can see symbols for the EPA safer chemical ingredients list. So that's that green circle here if this is a compound on there, as well as our greener alternative product symbol for things that are commercially available from Sigma Aldrich. So this is a program that we have within Sigma Aldrich that shows compounds that are part of the greener alternative product symbol, have this green symbol on it with the arrow and the globe, which means these products have been developed to fall into one of these 4 categories of whether they've been specifically engineered to reduce the environmental impact or aligned with the 12 principles of green chemistry, for example. So you see these symbols not only on our Sigma Aldrich website, but also directly in Cynthia. And then if we look now at the molecule details for any of the compounds in Cynthia, you see those greener alternative product symbols, as well as chemical hazard symbols and links to PubChem where you can get more information about any hazards or considerations there. So all of this really gives you information that you can use to kind of inform your synthesis and ideally plan something that's going to be greener or more sustainable. And then beyond that, we can also customize our pathways for more specific goals. If there's a specific hazardous reagent that we are looking to avoid, for instance, we can give it that information or promote certain kinds of chemistry, maybe enzymatic chemistry that's likely to be greener. And so you can build all of this in. And so to highlight that, I really want to go through a few case studies, which are real world examples where we actually used Synthia to optimize routes to some of our products. So these are coming from our lab in Bangalore. All of our chemists have access to Synthia to use in their daily work. And so these are just a few recent case studies that I want to share here. So I'm going to go through 3 compounds, 6,4-malterin, this lithium chromo ionophore, and this 5-fluoromethoxypyrimidine carboxylic acid. So to get started, the first one is the 6,4-malterin. This is a heterocyclic compound. That's the result of the photodegradation product of folic acid. It's implicated in various disease states, and it was a project we were looking at synthesizing. There is a reported one-step synthesis directly from folic acid, but unfortunately this was not reproducible in our lab, so we then looked for alternatives routes. There is another published synthesis that I have shown here from the Journal of Agricultural and Food Chemistry. You can see it is a long synthesis at 8 steps, and there were a few other issues with it of why we really didn't want to follow this. So in addition to one just being a long synthesis, it started from this compound that was very expensive. It was over $600 a gram, so that was going to be a challenge. The first step is using this toxic reagent PCl3, so ideally we were looking to avoid that. And then overall had low atom economy, so ideally wanted to improve that. So you'll see these the second and third steps, which are transferring out, first adding the pyridine and then the dimethylamino nitrobenzene, which are later removed. So it's not very atom economical. So using Synthia, they were able to develop now this 4-step route that addressed those issues. Of course, we've immediately cut the number of steps in half, so that's an improvement there. And really we have the key step here is this selenium oxide oxidation of the methylpterin that Synthia proposed based on literature report. We could prepare the methylpterin precursor in one step from a much less expensive starting material. And then, we do have protection of the amine with an acetyl group, and then removing that later, but overall is still a lot more atom economical than the original literature route. So if we look at kind of a summary here, we were able to reduce the number of steps, improve the overall yield from 16% up to 28%. We also greatly reduced the cycle time, so that's the time that it takes to, actually do all of the reactions and purification, from 10 days down to 4 days. This also significantly reduced the labor costs, and then the raw material costs were also really significantly reduced from $7,600 down to $120 for those initial raw materials. So this led to an overall cost savings between the labor and the raw materials from almost 12,000 US dollars down to 2,600. We also took a look at the green metrics here. So we have a tool called Dozen that we use, which is a quantitative measure of the greenness of a process and how well it aligns with the 12 principles of green chemistry. Essentially, the lower the Dozen score, the greener the process. And we compared the initial literature route to the Synthia route, and we were also able to see a nice improvement there in that Dozen score, specifically looking at some things of it being more atom economical, having less hazardous chemicals, and less waste. So the next project that I want to talk about is this lithium chromoionophore. This is an endo-aniline dye that can selectively complex ions to cause a change in absorption and emission properties, which has potential applications in trace metal detection, in biological systems, as well as for molecular data processing. This was also something that had been synthesized previously, so I have shown the reported 6-step literature synthesis here, which was initially used in our lab, but the overall yield was quite low, and so wasn't able to produce enough for the batch that was required. So this was another one that we wanted to optimize. One of the issues here was this intramolecular cyclization. Step 3 was especially low yielding. So when we used SYNTHIA, was able to propose now a 3-step route. And what was kind of key here is that SYNTHIA recognized that we could actually start from a commercially available compound, the azocrown ether, and directly couple that to that fluoronitrobenzene in the first step, avoiding having to do that low yielding cyclization. This allowed us to improve the overall yield up to 15% and produce enough material here. This also improved the overall atom economy of the route as well. So again, taking a look at this summary here, reduced the steps from 6 down to 3, essentially tripled the overall yield, again, reducing the cycle time and the labor costs. In this case, the raw materials were more expensive than with the literature route, but because of the savings in labor cost, it still reduced the overall cost by about half. And then my last example here, this is another compound coming from a recent MedChem project. In this case, the published route from this patent was 2 steps, so not especially long, but the key issue was that we wanted to avoid using the hazardous cyanide reagent in that first step. So again, looking at SYNTHIA, was able to find an alternate 2-step route that avoided the use of that chemical. It also avoided the expensive raw material that we needed in the first literature procedure. So we were able to reduce the overall costs and also had a slight yield improvement. So again, taking a look at the summary here, was able to improve the overall yield from 29 to 34%. Also reduce the cycle time and labor costs as well as raw materials for about a 50% cost savings there. And so if we kind of summarize all of this together, with these projects, we were able to increase yields, reduce cycle times, as well as overall costs, and improve those green chemistry metrics for these compounds, by using SYNTHIA to help us with that route optimization process. And so with that, thanks for your time. If you're interested in trying SYNTHIA, you can access it, a free trial version at light.cynthiaonline.com. And again, thanks for the opportunity to talk today.